The next test is called the complement fixation test. Now this test is a little bit old school and we don't really use it that much anymore, but the textbook thought it was important enough for you to know about, so you have to know about it. So here's how this test works. Now in this picture, these top boxes are showing you a patient that's positive for a disease, and these bottom pictures are showing you a patient that's negative for the disease. So what you would do is you would take the antigen, a known antigen, and you would put it in a test tube. This could be whatever you want, make it up. It could be coronavirus, flu, Staph aureus, whatever. Okay, so you put the viral antigen in there. Then you add the patient's serum. If the patient has antibodies to this antigen, if they're positive, then the antibodies and antigen will attach to one another. If the patient is negative, they do not have any antibodies against this antigen, of course there's not going to be any complex. The antigens are still free. Next, we add this substance called complement. Complement are proteins that are made by the immune system naturally, and complement always will stick to antibodies that are bound to antigens. So in this case, this person has antibodies bound to antigens, so the complement will attach, will fix itself to that antibody. But in this bottom picture, there's no antibody that's complex with the antigen, so the complement is still free. Now the next thing we do is we add sheep red blood cells, and we also add additional antibodies called lysins. Lysins will attach to the sheep red blood cells. They're basically just antibodies that lyse red blood cells. So we add the lysins, so both tubes will get the red blood cell with the lysins. Now in the last step, this patient that has antibodies, the complement is bound to it, so complement cannot bind itself to the red blood cell, so there's no hemolysis. So the end result, if the patient's positive, you're going to see the red blood cells are still red, they're still intact. This other patient down here at the bottom, they do not have any antibodies. Remember, complement is still free. What's going to happen is, remember, complement likes to bind to antibodies, so the complement is going to bind to the lysins here, and it's going to cause them to lyse the red blood cells. So you're going to see hemolysis. So in this case, if the patient is negative, you're going to see hemolysis, meaning the red blood cells will lyse, and the test tube is going to turn from red to maybe like a pinkish color or like a light color because the red blood cells are breaking open. So in the end, again, positive result, meaning the patient does have antibodies. There's going to be no hemolysis. Negative result, meaning the patient does not have antibodies, then you're going to see hemolysis. So the results are kind of the opposite of what you would think. Positive, no hemolysis. Negative, hemolysis. Now, there are some miscellaneous serological tests that are used for, again, specific diseases. So I'll briefly tell you about them. The Treponema pallidum immobilization test. This is to test if somebody has antibodies for syphilis. So how this works is, remember, syphilis has that corkscrew shape and it uses periplasmic flagella, so it's mobile, it's always moving like a corkscrew. If somebody does have antibodies against syphilis, what would happen is we take the patient's serum and they had antibodies, then these antibodies would actually stick to the syphilis bacteria and prevent the bacteria from moving, so it would immobilize the syphilis. So that's one test we might try. Another random miscellaneous serological test is the toxin neutralization test. So remember, a lot of bacteria produce exotoxins that can cause very serious diseases. So if we want to see if a patient has had a disease before in their life that produce an exotoxin, then the patient will likely have antibodies against these exotoxins. Antibodies against exotoxins are called antitoxins. So basically what we would do is we would take the known bacteria that makes the exotoxin, put it in a petri dish, grow it, and then we would add some of the patient's serum. If the patient's serum killed off the bacteria, we know that the patient is positive. They have antitoxins. If the bacteria can continue to grow, then they're negative. They don't have the antitoxin. Serotyping is used to identify specific strains or serotypes of diseases. An example, a classic example, is the quelling test. Remember that I told you before that Streptococcus pneumoniae, which can cause pneumonia, meningitis, ear infections, etc. It has many different types of capsules, and each different types of capsule makes the strain a different serotype. So what we could do is if we suspect that somebody has strep pneumonia and we want to find out which strain, which serotype, we could extract some of the strep pneumonia and test it against different antibodies that will attach only to specific capsules. And when we see the positive result, usually indicated by swelling, that's why they call it the quelling test, because it kind of looks like the bacteria is swelling, but it's actually just that the antibodies are attaching themselves onto the capsule. And that's how we can figure out which strain of strep pneumonia a person had. Lastly, there are some in vivo tests that we can use that are serological. So for example, the tuberculin skin test, we actually will inject a little bit of the antigen, a little bit of the protein from the Mycobacterium tuberculosis. So we're not injecting like live bacteria, we're just injecting a little bit of the protein from the bacteria into the skin. And then we wait about 48 to 72 hours. And then we look to see if there is a reaction, and if there is, that means the person probably has antibodies against Mycobacterium. It doesn't tell us if they're actively infected, but it can kind of give us an idea that they have been infected at some point in their life, or they may have a latent infection. The next test to discuss is immunofluorescent testing. So remember I told you that antibodies and antigens bind, and we need a way to be able to visualize if they bind or if they don't. So another way that we can visualize is using fluorescence. In this case, we would need a fluorescent antibody. So basically, we're going to need some sort of antibody, and we are going to have to add a fluorescent tag to the antibody. And we add the fluorescent tag to the stem region of the antibody. This is also known as the Fc region of the antibody, because we need the arms to still be able to bind to the antigen. Now, fluorescent antibodies that have these fluorescent tags, these are types of monoclonal antibodies. Mono means one. So sometimes if you have, for example, polyclonal antibodies, that means you could have like a single pathogen or a single antigen, and there's like many different types of antibodies that will bind to different antigens on the surface of the pathogen. So like maybe some of the antibodies bind to the pili, maybe there's some antibodies that bind to the flagella. So there's multiple antibodies that bind to multiple antigens. But in terms of a monoclonal antibody, that means that there's an antibody that binds only specifically to one specific antigen. So again, monoclonal antibodies are highly specific. Now there's two different classes of immunofluorescent testing. The first class is direct testing. And we're going to talk about many direct tests, like direct immunofluorescent testing, direct ELISA. Here's how direct testing works. Let's say that you have a patient that you suspect maybe has one of these diseases, because we use direct testing to diagnose syphilis, gonorrhea, chlamydia, whooping cough, Legionnaire's disease, plague, a lot of different diseases are tested using this. So for example, let's suspect that you think you have a patient that has gonorrhea, right? And you know Neisseria gonorrhea is a Diplococcus-shaped bacteria. So what you would do is you would take the patient's specimen that you suspect has the gonorrhea bacteria in it, but you don't know. And so you have this basically unknown antigen. Then what you would do is you would buy monoclonal antibodies that are commercially available that are very specific to Neisseria gonorrhea. So if this patient does have gonorrhea in their sample, these monoclonal antibodies will bind to the surface of the gonorrhea bacteria. And remember, there's a fluorescent tag on these antibodies. Then you would expose the sample to ultraviolet light. And so if you see fluorescence, then you know the patient is positive for gonorrhea. And if you don't see fluorescence, they're negative. So the reason this is called direct testing is because you're directly attaching the antibody with the fluorescent tag to the unknown antigen. It's very simple. The second type of immunofluorescent testing is indirect testing. Indirect testing is very similar, but there's a couple differences. So in indirect testing, instead of taking an unknown antigen from your patient's sample, instead you're taking a known antigen. Again, this antigen could possibly be the Neisseria gonorrhea, but this time you're taking a known Neisseria gonorrhea from your lab. Then you're placing the patient's serum. and the patient's serum contains unknown antibodies. So it's basically the opposite of the direct testing. In the direct testing, you had an unknown antigen and known antibodies. In indirect, you're using a known antigen and unknown antibodies. If the patient has antibodies against gonorrhea, probably indicating they are currently infected, these antibodies will attach to the bacteria. If the patient does not have any antibodies, then obviously nothing will attach. Now, indirect testing requires a third step. This is what makes it indirect. You now have to add a secondary antibody. The secondary antibody is the one that has the fluorescent tag on it. The secondary antibody, the arms of it, will bind to the Fc region of a Neisseria gonorrhea antibody. So if the patient is positive for gonorrhea and their antibodies have bound to the bacteria, then the secondary antibody will bind to the primary antibody. However, if the patient was negative, like in this case, then the antibody won't have anything to bind to and it'll just kind of be floating around in the sample. And then once again, we will expose the sample to ultraviolet light. If the antibody is bound, as in this case, the patient's positive, you will see fluorescence. And here's a picture of some bacteria with the fluorescent antibody attached to it, so you will be able to see it. So indirect immunofluorescent testing is often used to diagnose syphilis as well as a variety of viral infections.